Good morning, everybody. So now today we're going to start with meiosis. Last period we finished with mitosis. We discussed in detail what is mitosis, where it occurs, and what are its significance. Now we'll have just a recap of it. Mitosis occurs in somatic cells, meaning it occurs in all the body cells except the reproductive organs. Now, what are the significance of mitosis? It helps for regeneration of damaged cells. It helps for healing of wounds as well. And in asexual reproducing organisms, it is a method of reproduction. One cell divides into two cells where the number of chromosomes remain constant. It does not change. When one cell divides to form two cells, both the cells which are formed, which are called as daughter cells, they both have exactly the same number of chromosomes as that of parents. So there is no genetic variation, no differences in the new cells which are formed. That is the speciality of mitosis. We also discussed that the entire process of mitosis is divided into two stages or two steps as karyokinesis and cytokinesis. Karyokinesis again divided into four stages as prophase, metaphase, anaphase and telophase. Then we have second stage, that is cytokinesis, where the cytoplasm divides and finally two daughter cells are formed. We discussed what happens in prophase. Prophase is the stage where the chromosomes start forming from chromatin material. The thread-like DNA starts becoming compact and finally the chromosomes are formed. Then slowly the nuclear membrane start disappearing, centrioles start reappearing, centrioles start dividing and they start moving to the opposite ends. This happens in prophase. Second was metaphase. In metaphase, the centrioles have shifted to the opposite ends, spindle fibers are formed and all the chromosomes are aligned in the equatorial position or in the center of the cell. This happens in metaphase. The spindle fibers have been attached to the centromere. Next stage is anaphase. In anaphase, the spindle fibers slowly start pulling the chromosome towards the opposite ends, and hence each chromosome which was having four strands or four arms, it gets split. Two arms on upper side and two arms start going on the opposite side. So entire chromosome gets broken up into half. That is, the four strands or four branches divide to form two branches. Two branched chromosome moves to the one end and the other half moves to the other end. This starts happening in anaphase, and finally in telophase, the chromosomes, the half divided chromosomes have reached to the opposite ends. Nuclear envelope starts reappearing again and the chromosomes start again decondensing, or they again form the thin thread-like DNA. This happens during telophase. Next, cytokinesis. Now once this telophase is completed, we have one cell with two nucleus. During cytokinesis, the cytoplasm starts furrowing and finally the furrow deepens and two cells are formed with exactly equal number of chromosomes. In short, this was mitosis. Now let's start with meiosis. Meiosis occurs in gametic cells or in germ cells only. That is in case of humans. Meiosis occurs in males in testes and in females in ovaries. Now this is a very special kind of cell division, because here the chromosome number is reduced to half. Now let's take our example. We have 46 chromosomes in each and every cell. 46 chromosomes means we have 23 pairs. Like I have a pair of shoes, that means I have two shoes. I have two pair of shoes means I have four shoes. Same way, when I say 23 pairs, that means each pair contains two chromosomes. So like that, 23 pairs, that is 46 chromosomes in total. Now during meiosis, this 46 chromosome cell divides in such a way that each daughter cell receives 23, that is half the number of chromosomes, that is haploid. What is diploid, haploid we studied in last period. Now let's start in detail about meiosis. Now meiosis is a very lengthy process and occurs less frequently as compared to mitosis. In meiosis, it is divided into two stages as meiosis 1 and meiosis 2. Meiosis 1 again has the stages as prophase 1, metaphase 1, anaphase 1, telophase 1. Same thing goes with meiosis 2 also. Now meiosis 2 again has prophase 2, metaphase 2, anaphase 2 and telophase 2. And finally cytokinesis. In meiosis 1, prophase 1 is the longest process. Prophase 1 is divided again into 5 stages as leptotene, zygotene, pachytene, diplotin, dikinesis. These are the 5 stages in which meiosis 1 is divided. Now what happens in leptotene? Leptotene means chromosomes are thread-like. Now here we have to remember that homologous chromosomes, they start coming towards each other. Now in a cell, when cell is not dividing, the chromosomes are spread everywhere or the chromatin is spread everywhere. Now once the cell decides to divide by meiosis, the homologous chromosomes which are scattered everywhere, they start coming or they start finding their pairs and they start coming towards each other. Suppose chromosome number 1, it is in pair. So one chromosome number 1 is somewhere else and the other chromosome number 1 is somewhere else. So they start coming or they start finding each other. So like this 23 pairs start coming or coupling together. Like we have entire shoes placed together and then we start sorting them with one another, one pair with other. So that is how the chromosomes start finding each other. The chromosomes start pairing. That happens in leptotene. Second is zygotene. In zygotene, once the homologous pairs of chromosomes find each other, they attach. One of the arms start attaching to one another. That happens in zygotene. They get closely packed with one another. Next is pachytene. In pachytene, the arms of chromosome overlap on each other. That is called as crossing over. Now what happens during this crossing over? When the arms overlap on one another, the segments of chromosomes get exchanged. That is, they are in pairs. One of the arm of one pair overlaps or intersects the other pair and then it breaks at that point and attach it in such a way that one part of one chromosome is cut and attached to the another chromosome and vice versa happens. So small segments of chromosomes may get exchanged during this stage, that is pachytene stage. Now after pachytene, diplotene. In diplotene, the exchange is completed and in dikinesis, the chromosomes start going away from each other. So these are the stages. You can check or compare in the diagrams which I have posted next to the explanation. You just see the two chromosomes, homologous chromosomes are shown in two different colors. And when this process of zygotene, diplotene, dikinesis occur, you will see slightly one colored chromosome has exchanged its segment with the other color. So as a result, at the end you will see entire chromosome of different color with a small segment of another color. That other part is of the chromosome which was its homologue. So this is how exchange of genetic material, exchange of segments of chromosomes occur during prophase 1 and finally it is completed. Though it is completed, the chromosomes are still attached to one another at some points. And in this stage only the chromosomes move to metaphase 1. Now during metaphase 1, again the same process, all the chromosomes are aligned in the center. But the difference is here, the chromosomes do not get separated from each other. They are in pairs. So all the pairs get horizontally arranged and spindle fiber from opposite end attach only to one chromosome. Out of a pair, one spindle fiber attached to one chromosome and the other spindle attaches to another chromosome, which are attached to each other from prophase 1 itself. So here metaphase chromosomes align in the center, but they are in pairs, they are attached with one another. As a result, only each spindle fiber attaches with only one centromere of one chromosome, and other centromeres other centromere is attached by another spindle. So this happens in metaphase 1. Next is anaphase. In anaphase, the chromosomes are pulled apart. Now during pulling, what had happened in mitosis, one chromosome was split to form two chromosomes, but here in meiosis it does not happen. Suppose 23 pairs are lying in equatorial position, 23 pairs, then the spindles start pulling it apart, and out of 23 pairs, one pair moves, one chromosome of each starts moving in one end and another pair starts moving in another end. So this is the difference. What happens over here? Out of 46 chromosomes which are aligned in the equator, 46 chromosomes means 23 pairs. Out of that 46, 23 moves up and 23 moves down. So during anaphase, half the number of chromosomes start moving in one pole and half the number of chromosomes start moving in the another pole. Telophase, again the nuclear membrane starts reappearing, the chromosomes have reached to the opposite end, spindle fiber disappear, and then we have cytokinesis. But in meiosis, cytokinesis does not occur. Directly this cell with two nucleus moves to meiosis 2. In meiosis 2, again what happens? These two partially separated cells are formed. They are not completely separated from one another as shown in the textbook. They are partially separated, or slightly they are still connected to one another. Then in meiosis 2, again prophase 1 starts. Prophase 2 Prophase 2 is same as mitosis. That means again the chromosomes become thick, nuclear membranes start disappearing, spindle fibers start reappearing in both the partially separated cells, and again it moves into meiosis 2, metaphase 2. In metaphase, again each cell containing 23 chromosomes align in the center in both the cells or both partially separated cells. Then again anaphase, they start pulling towards opposite ends. Now here 23 chromosomes separate in such a way that each end receives 23. Each chromosome is a tetrad. Each chromosome now has 4 arms. So 2 arms start moving on the opposite end and the other 2 arms on the other opposite end. So it is exactly same like mitosis. Here 23 chromosomes finally divide in both of the cells to form 23 chromosomed cells again. So metaphase, chromosomes align in the center in both the cells. Then anaphase, 23 chromosomes divided into half. They start pulling to the opposite end. Telophase, they reach to the opposite end. Again nuclear envelope is formed, and finally cytokinesis begins, and again from each cell two cells are formed. So you can conclude that from meiosis, one cell divides to form 4 cells. In meiosis 1, 2 cells are formed, and those 2 cells again divide to form 2 more cells. So finally one cell divides to form 4 cells, and each cell contains 23 chromosomes. That is half the number of chromosomes. 46 chromosomes were there, they divided to form 23 chromosomes each, or 23 pairs were there, and finally when 4 cells are formed, they only contain one set of chromosome, or only 23 chromosomes. That was about meiosis. Now meiosis occurs during reproduction or during formation of gametes. In males, meiosis occurs for formation of sperms, and in females meiosis occurs for formation of egg or ovum. So that was meiosis. Now in the end, in your book there is a small activity given. That activity you will do in lab. It is simple. Onion roots we have to understand mitosis in onion roots. That experiment we will do in lab. You can just read it for your information, but that will be taken once we actually begin with the physical classroom. So this was end of lesson. You can start completing the exercise for it.